Welcome back to BioNerds and Engineers. I'm your resident biologist, mapping the code of life. And I'm the engineer, scaling up biological designs into industrial hardware. Today, we are looking at the ultimate planetary cleanup project. The Earth is currently facing a massive material backlog. Every single year, human activity generates over 390 million tons of plastic waste, running in parallel with skyrocketing industrial carbon emissions. Nature actually has biological countermeasures for this. Certain naturally occurring bacteria, fungi, and algae already possess metabolic pathways to absorb carbon and consume plastics. From an engineering perspective, the immediate question is throughput. If we drop a standard PET water bottle into a vat of these naturally occurring plastic-eating microbes, how long does it actually take for them to break it down in the real world? Weeks or even months? Natural microbes are incredibly sensitive to temperature and pH changes. Worse, when they attempt to digest large volumes of plastic at once, the chemical byproducts build up inside the cells and literally poison them. Nature gave us a working prototype, but to clear hundreds of millions of tons of waste, that biological machinery requires a serious mechanical and genetic upgrade to hit industrial scale. This infographic outlines synthetic biology's major tools. Utilizing CRISPR, scientists rewrite the genetic code of living organisms, optimizing functions at whole cell, metabolic, and enzyme levels. For example, editing instructions for the Rubisco enzyme allows algae to fix carbon significantly faster. But plastic is a far more complicated chemical structure. If a single bacterium is metabolically poisoned by the byproducts of its own food, how do you scale the degradation process? Researchers at the University of Illinois and MIT bypassed the single-cell limitation entirely. They built a microbial consortium, a carefully engineered bacterial team operating on a strict division of labor. Instead of trying to construct one organism to do everything, they treat living cells like an industrial assembly line, assigning highly specific, isolated tasks to completely different engineered strains. PET breaks down into two toxic chemicals, terephthalic acid and ethylene glycol. If a natural bacterium attempts to consume both, metabolic pathways conflict and the cell crashes. To fix this, researchers hyper-specialized one strain to consume only the acid and engineered a second for the glycol. In computer science, we call this parallel processing. By dividing a massive computational workload across multiple cores, you prevent the entire system from freezing. Here, researchers are executing that exact architecture inside living cells. Because these two strains share the metabolic burden without interfering with each other, this engineered duo produces 92% more upcycled biodegradable polymer than a single strain attempting the same job. That 92% efficiency jump provides the necessary margins to move these biological systems out of academic research labs and into large-scale commercial facilities. Startups like LanzaTech are currently deploying massive bioreactors filled with customized, AI-monitored microbes to capture raw industrial emissions directly from the source. Those microbes physically ingest the toxic waste gases and output highly valuable liquid resources, specifically sustainable aviation fuels and industrial ethanol. To accelerate this timeline, companies like Ginkgo Bioworks use AI-driven biofoundries to automatically design, build, and test thousands of custom microbial variations, rapidly identifying the most efficient degradation pathways. We are reaching a point where humanity no longer has to simply bury or burn its chemical waste. By editing the genome, we are actively harvesting that pollution and transforming it into high-value commodities. By treating toxic carbon and ocean plastics as the raw feedstocks for tomorrow's materials, we achieve true closed-loop engineering. Thanks for tuning in to BioNerds and Engineers. We'll catch you on the next build.